This video is going to be on the mobilization of fatty acids. So, for us to mobilize fatty acid for other cells to use, is going to be we're going to actually go to the site where it is stored. So, fatty acids are stored in form of triglyceride in adipose tissue. That is where fatty acid is stored. Fatty acid is stored. The major site for storage for fatty acid storage is the adipose tissue. So we are going to pick an adipocyte. We're going to pick an adipocyte. So when we pick this adipocyte, we are going to break down the triglyceride into glycerol, glycerol, and fatty acids. So the process of breaking down these triglycerides into what we call fatty acid and glycerol is called lipolysis. The other question to ask now is, when does this lipolysis actually occur? When is it actually occurring? So the actual time or period that this lipolysis becomes so high is when there is low glucose level, like low blood glucose level, input blood, and during, let's say, sympathetic stimulation. When there's sympathetic stimulation, that means there's release of epinephrine. So when there's release of this epinephrine, then you have this lipolysis being favored. So now let's look at the adipocyte. So let's assume that this is an adipocyte. Let's make this the cell membrane of the adipocyte. So now at the adipocyte we have lipid droplets. So the lipid droplets, it assume this is a lipid droplet. It has a membrane. The lipid droplet has a membrane. So now on the lipid droplet membrane, there's a protein called perilipin. Let me use another color for that. This is the lipid droplet. So there is a protein called perilipin on the surface of the lipid droplet membrane. So now this protein perilipin interacts with another protein called CGI58. So this C stands for comparative gene identification. So it's a protein. So this CGI protein loves to interact with the unphosphorylated form of this perilipin. So it loves to interact with the phosphorylated form of this perilipin. So it interacts with this perilipin. So now on this same surface again, there is, let me just use blue. On this same surface again, there is an enzyme called adipose triglyceride lipase. Adipose triglyceride lipase is an enzyme that's involved in this lipolysis. So now there's an enzyme again, you guys are familiar with this one, that is located at the cytosol. We have what we call hormone sensitive lipase. That's the name of this enzyme, hormone sensitive lipase. So now I'm showing you all this so that when the hormones actually stimulate the receptors, you see the effect of this stimulation on this. Remember, this is an enzyme. Sorry, this is an enzyme. And remember, this is an enzyme here. This one is a protein. This one is a protein. So this perilipine, why you are seeing it on the surface, is to prevent these lipases from acting on this lipid droplet. So that's why this perilipine is there and interact with this comparative gene identification 58. Remember, I said that this comparative gene identification 58 is a protein. It's a protein. So now, I also told you that it loves to interact with the dephosphorylated form of this perilipine. So now we're going to bring the hormone to stimulate this process. So let's assume the blood glucose level is low. We need fatty acid. We need to mobilize fatty acid for it to be utilized by the metabolizing cell. So now we're going to use epinephrine. Glucagon can act on this, can also cause this process to occur, but it's more preferred I use epinephrine because glucagon receptor, they are primarily more located on the liver, cell, the hepatocytes. So now the beta, I'm going to use beta because the epinephrine are going to act on, for them to stimulate this process, they'll act on the beta adrenergic receptors. Oh god, this handwriting is ugly. So they're going to act on the beta adrenergic receptor, specifically beta 3. Sorry, specifically beta 3. So beta 3 is on the adipocytes. So, and it is what? G protein coupled. It's a G protein coupled receptor. So now, G protein coupled receptor. So now, let's draw this. Let me take it higher. So let's assume this is the receptor, right? And 1, 2, 3, 4, 5, 6, 7. I know the G protein coupled receptor is actually a serpentine. It goes 7 times on the cell membrane. Serpentine receptor. So now we're going to have an enzyme here. So this enzyme here is going to be called adenylyl cyclase. So this enzyme here is going to be called adenylyl cyclase. So I'll put ACL, adenylyl cyclase. So now the epinephrine will come bind to this. So this is the epinephrine. We represent it EP. So it binds with the beta 3 adrenergic receptor. So when it binds with this now, this G protein has subunits. You understand? It has subunits. So the subunits are alpha, beta, and gamma. So the alpha at rest is bounded to GTD. You understand? It's bounded to GTD. So they have this G proteins. They have GTPase activity. So, oh sorry, this is a mistake. It's going to be, it's not GTP, it's not GT, it's GTD, it's G, it's bounded to GDP, guanyl diphosphate, guanosine diphosphate. So it bind to guanosine diphosphate at rest. So at rest, it bounded to guanosine diphosphate. So since it's bounded to guanosine diphosphate at rest, so that means for us when this when this epinephrine binds, so GTP will replace GDP. So now GTP will be replaced by this. So GTP, the alpha will bind to it. So this alpha now, since it's bounded to this now, it will take this GTP to this guanylyl cyclase, give it one phosphate to phosphorylate this guanosine to phosphorylate this adenylyl cyclase. So this will phosphorylate the adenylyl cyclase, so the adenylyl cyclase will become active. So remember, if you give one phosphate, what do you think you have? You have GDP. So this alpha will take the GDP and go back to its normal form again. So back to its normal form. So now this adenylyl cyclase will now act on ATP. So if it acts on ATP now, converting it to what? Cyclic AMP. So since they say cyclic AMP, meaning that the phosphate, it's having a single phosphate now, is connected to the third and the fifth carbon of the ribose sugar. So it's connected to the third and fifth. So now this adenylyl cyclase now will now act on protein kinase A. To act on protein kinase A. So now it's protein kinase A that will now carry out the remaining reaction now. So now let's now focus on protein kinase A. Let's focus our attention on protein kinase A. So focus our attention on protein kinase A. So we had protein kinase A. So protein kinase A will now phosphorylate hormone sensitive lipase. So phosphorylate hormone sensitive lipase, and I showed you the lipid droplet that perilipene is there, and perilipene is bounded to who? The CGI58 protein. So remember, it loves to bind to the dephosphorylated form of perilipene. So now remember, I told you there's an enzyme too, adipose triglyceride lipase, right? So now this protein kinase A should phosphorylate this hormone sensitive lipase and also phosphorylate perilipene. So since perilipene has been phosphorylated now, this comparative gene identification 58 will now separate from the perilipene because, remember, because what? It loves to bind to the dephosphorylated form of perilipene. So now since that we separated now, so it will leave it. Then it will now, this CGI58 love to activate adipose triglyceride lipase on the cell membrane of this lipid droplet. So when it activates this adipose triglyceride, so remember I told you the CGI58 that activates the adipose triglyceride. So this adipose triglyceride is the first enzyme to act on triglycerides. It's the first enzyme to act on triglyceride. It has high affinity for triglycerides, while this hormone sensitive lipase has high affinity for diglycerides. You can also call it diacylglycerol, and you can also call this TG as triacylglycerol. So it loves to act on diacylglycerol, and it's another enzyme again that will be activated, which is also called monoacylglycerol lipase. So this monoacyl glycerol lipase will be also be activated. So they have not how will I put it now? They're still actually they're still finding the actual way these enzymes actually perform their actions. So not everything is actually clear on how they actually carry out their actions. So they've gotten just some just little information about the way these enzymes actually perform their action. So but I'm going to show you how they actually perform some of their actions. So now since I've showed you that this is what how they are activated, so I'll show you. So if you look at this structure now, this is triglyceride. This is triglyceride, or you could also call it triglycerol. So this triglyceride is acted upon by lipase. So this lipase represents adipose. Okay, adipose. It represents adipose triglyceride lipase, and also it could be hormone sensitive lipase, and it could be monoacyl lipase. So, but for now, I told you the first person to act is acyl triglyceride lipase. It has been like for a long time, we've always been thinking that it's hormone sensitive lipase that's responsible for all the actions. Like it totally breaks the triglyceride down. But actually, that's not the case. It's actually respond to hormone. This hormone sensitive lipase, yes, it regulates this because it responds to hormone. But the first enzyme to actually act on triglyceride is adipose triglyceride lipase. So when it acts, it removes one fatty acid. So now let's assume after this um, adipose triglyceride lipase act, remember if it's acting now, it adds water. Let's break water down to hydrogen and hydroxyl ion. So if I give hydrogen to this bond here, the whole of this structure here, you now have a fatty acid. So this R is for alkyl group. It could be any number. Maybe it could be 4 carbon atom, 6, 8, so and so. So after adding, sorry, I'm not adding hydrogen there. I'm not adding hydrogen there. Let me clear this first. I'm not adding hydrogen there. It will be correct if I add hydrogen there. So what I'm going to add is this is OH, this is hydrogen, right? So I'm going to add OH to this structure. Then we'll get the fatty acid. So I'll now take the H and add it here. Then we'll get an hydroxyl group. So now remember this is the action of who? We can't go into detail. I told you that the actions are not really clear. So I'll just take on the ones that are the actions that are more well explained and will be well understood. So now this adipose triglyceride perform its action. So go to hormone sensitive lipase. Remember I told you that it has high affinity for diacyl glycerol or diglycerides. So when it acts on this diacyl glycerol now, the same thing, add hydroxyl group, you form fatty acid, then add hydrogen, you have OH. So if monoacyl glycerol comes, it has high affinity for monoacyl glycerol. So when it add hydrogen, when it add OH, another fatty acid, then add hydrogen, what do you have? That's it. What do you have now? You now have a glycerol. Then you have you actually removed 3 fatty acids and you had 1 glycerol. So now let's see how these fatty acids and glycerol are going to be transported from the adipose tissue. Let's go back. So now we're going to assume that and make a straight line. This is adipose tissue. So we're going to assume that this is adipose tissue and this is the blood vessel. There's a capillary. There's a capillary. So, and we actually synthesize glycerol. Okay, let me use GL to represent glycerol. And we actually synthesize fatty acid 3. Not synthesized, we actually broke down triglycerol to generate fatty acid. Yes. So now there's a transporter named aqua aquaporin 7 that is present on the cell membrane of the adipocyte. So this aquaporin 7 transports water and also uh, allow the outward movement of this glycerol. So it allows the transport of glycerol from the cell. So this glycerol will be transported from the cell by this aquaporin 7 to the where? To the blood stream. Right? So if we enter the bloodstream, which part of the bloodstream do you think it will enter? The venous system actually. So because the arterial system is supplying the cell, so the venous will take it to the heart and to be pumped to everywhere where it is needed. So now the same thing with this fatty acid. Fatty acid will be transported by the fatty acid transporter protein. Fatty acid transport protein, sorry. So if fatty acid transport protein will transport the fatty acid into the bloodstream, same thing. Everything will take into venous system. Venous system will take it to the heart and it will be pumped, taken from the heart, taken to the lungs to be oxygenated, then pump into we'll take it to the uh, left atrium, left ventricle, and you're pumped into the aorta and distributed into uh, systemic circulation. So now, after we have actually been able to transport the fatty acid into the bloodstream. So in the bloodstream, the fatty acid has a transporter. Yes. So there's a transport protein for this fatty acid because the fatty acid is not actually compatible with the plasma because the plasma is made up of 99% of or 90 something percent of water or 99% of water. So now, so since it's not that compatible with uh, the plasma now, so there's a plasma albumin. So plasma albumin has 7 binding sites. It has 7 binding sites for fatty acids. So and you know that this we are only taking on one adipocyte now, not plenty adipocyte. So this same process is happening in all the adipocytes in the adipose tissue. So they are releasing fatty acid into the bloodstream, and the albumin are actually binding this fatty acid. I told you that the albumin has 7 binding sites for these fatty acids. So they'll transport the fatty acid to the cell that need this fatty acid. So let's say we have been able to actually take it to the blood. You know, since it's through the venous blood, so if it's enter the venous blood needs to be oxygenated. So if it's oxygenated, it will be taken to the left atrium. From the left atrium, then left ventricle, and before the left ventricle can actually pump it into the systemic circulation. So let's assume it has done that. So and this is a cell. Okay, let me clean this. Sorry. And this is a cell. This is a cell. Let's say a liver cell. Yeah, let's say a liver cell that is in need of this fatty acid. Let's say a liver cell is in need of this fatty acid. So remember, in the bloodstream we had the fatty acid bounded to albumin. Remember, albumin has four seven binding sites. Then glycerol too. So this glycerol can either go to the liver or the kidney. So, but we'll take this glycerol to the liver. So this glycerol will be transported into the liver by an aquaporin protein. So the aquaporin protein now is called aquaporin 9. So that's the one present on the liver cell that will transport the glycerol into the liver. So if this glycerol enter the liver, it can be acted upon by who? It can be acted upon by glycerol dehydrogenase. So if it's acted upon by glycerol dehydrogenase, to be converted to what? To glycerol dehydrogenase. Oh, sorry, not glycerol dehydrogenase, glycerol kinase. I'm sorry, it's a mistake. It's glycerol kinase. Glycerol kinase. Let me use GL to represent glycerol. Glycerol kinase will act on glycerol and convert it to glycerol phosphate. Glycerol 3 phosphate. Sorry, glycerol 3 phosphate. So this glycerol before that glycerol 3 phosphate before that acted upon by glycerol dehydrogenase to produce what we call glyceraldehyde 3 phosphate, which can be channeled into glycolysis or be channeled into generating glucose. You know, gluconeogenesis is taking place in the liver. So now, actually, how this complex here, this fatty acid complex here, is actually being broken and transported, and the fatty acid transported into the cell is actually not known. It's not actually clear yet. So now we just assume that the fatty acid will be transported into the cell. So if it's inside the cell now, it's transported into the cell by what? By a fatty acid transport protein. So it will be transported into the cell by a fatty acid transport protein. So in the next video, we're going to activate this fatty acid. We're going to activate this fatty acid so that it can undergo what we call when it's activated. Actually, after activating it, maybe I'll release another video on beta oxidation.